pleasure to be here. It's my first time to this institute, so I'm really looking forward to meeting many of you and having a wonderful day. So I'm going to talk about using big data to disrupt the healthcare system. So I believe that the healthcare system, at least in the United States, is broken. We really practice sick care rather than health care. And even when we practice health care, it's very antiquated what we do. Basically, what we do is we typically get in a car, always at an inconvenient time, travel to a physician's office. It really hasn't changed much in the last 40 years, a few new gadgets. And then while you're there, they'll stick a needle in you and take a lot of blood. And from all that blood at the time you're there, they actually don't make very many measurements. Let me get out of your way. And then from those measurements, they'll treat you based on population averages. We think every one of these steps can be improved. And so specifically with regard to this last step, at least in the United States, we're told since you're little, your temperature, sorry, I mean, I have to convert over, about 36.4 centigrade, 98.6 Fahrenheit. If you actually look at the data though, it's really more like this, 97.5. In fact, there's just a big study out of Stanford, 300,000 people had 97.7. But the more important point here is there's a spread. This is the 25th quartile, 94.6. That's a 75th, 99.1. And so what that means in today's world is this is your normal healthy baseline at 94.6, and they measure you at 98.6 in the physician's office. They'll tell you you're normal, you're healthy, everything's fine, what are you doing here, go home. But I guarantee if you're up 4 degrees over your baseline, you're not healthy, something's off. And that's a big theme of our work, measure people's healthy baseline to see shifts from that. Now you probably know your health is influenced by many things. It's influenced by your DNA, which accounts for about 16% of your lifespan. And then all these other things will affect your health span as well, and lifespan. Activity, food you eat, mental health, environmental exposure. We know intuitively these will impact your health. But we're in a world where we can now measure all of these stuff quite well. We'll talk about wearables in a minute. Food logging still is measured indirectly. We're actually doing a lot to measure exposures with personal exposometers, actually, where we're trying to measure these. And even if you don't measure these directly, you can measure their effects by doing deep data profiles on people. That's thanks to revolution in technologies that's emerged. You probably know you can now sequence a genome with some of these new technologies. It costs about $100 in reagents. The interpretation is more than $100, so it's possibly up to several $100, depending on how deep you go. But the point is that this is now very affordable. Just out of curiosity, how many of you have had your genome sequenced, 23andMe kind of thing? Not so popular over here? Really? One. All right. Okay. Well, in the United States now, at least for getting genotype, it comes up quite a bit. So it's becoming very popular. In addition, so this gets a lot of attention, but you may know there's been a revolution in mass spectrometry, I would argue, over the last 15 years. We can now measure thousands of molecules on people's blood and urine, other fluids, and then the wearables, which I'll talk about in a minute, with smartwatches and continuous glucose monitors. So with all of these technologies, we can now do deep data dives on people. And so a number of years ago, it's basically 14 on me, I'm the first participant. But now, it's roughly 11 years ago, for a smallish group of people, 109, we've been profiling them very, very deeply. So we take their blood, urine, even their poop, their microbiome, and we'll sequence the genome once, and then out of their peripheral blood monocyte cells, their immune cells, measure the epigenome, mostly methylation, transcriptome, proteome. Out of plasma, we'll measure proteomes, cytokines, metabolites, lipids. Microbiome, we measure 4 different ones, gut, nasal, tongue, and skin. With collected samples for urine that will trick you. It's on top of deep clinical tests, questionnaires, some advanced tests like stress echocardiograms, glucose control measurements like insulin resistance, and other things as well. And then wearables, we started about 10 years ago, back when these were just fitness trackers. We thought they were powerful monitors. We've incorporated those in their cohort. So one aspect is to collect very, very deep data on people. And the other aspect is to do it longitudinally. So we sample people every 3 months while they're healthy. And then if an adverse event like a viral infection comes along, we'll take 5 to 7 more samples. So you might say, why are we doing all this? We're trying to understand what a healthy profile looks like, not a sick profile, a healthy profile. How does it change over time? What happens at the earliest times of illness? What's the difference between people, meaning what's the difference between you and the person sitting next to you? And then from the health perspective, can we use these advanced technologies to better manage people's health? And I can tell you when we started this, physicians are not like to sequence the genomes of healthy people. They thought we would turn everybody into hypochondriacs. It would cost us just millions of dollars. And many were quite outraged we were doing this. But now they've warmed up to it because they realize you can learn some very useful things from a genome sequence from this prediction. So with regards to this last point about using advanced technologies to manage health, what we discovered from just the first 3.5 years is that there were 49 major health discoveries. So nearly half the people learned something pretty important about their health, and it spanned a wide range of areas: hematology, cardiovascular, metabolic. And all of these were found pre-symptomatically, all these cases. And some of them were a pretty big deal, meaning we caught somewhat early lymphoma. These are pre-cancers that can convert to aggressive cancer, so those folks will get screened more. 2 people had serious heart issues, one picked up by genome sequencing, another actually by wearables, and so on and so forth. So we actually found these, once again, all pre-symptomatically, and no one technology did it. Sometimes it was genome sequencing, sometimes it was imaging. Often it was multiple markers, like lymphoma was both imaging and some biomarkers. And so these are actually from the profiling, the biochemical profiling. So again, the myriad of different technologies gave a much clearer picture of what's going on. The way we like to think about this is that if your health is a 1,000-piece jigsaw puzzle, we're trying to collect, say, 500 or 600 pieces in a much clearer picture versus 5 or 6 we would argue today. We just don't get very good data on people, and so we can see a lot more. So just to go through this in a little more detail, genome sequencing turned out to be quite powerful. Just in the first 70 people, we had 12 that learned something important from their genome. The ones in red are what are called the American College of Medical Genetics. The other ones were from other things we found in the literature. And it turns out that these are single gene mutations that put people at high risk. So you're probably all familiar with BRCA gene mutations, which put women at high risk for breast and ovarian cancer. This individual actually was an orphan, and so didn't know the parents, which was actually kind of useful for them. There were 3 cases where there were phenotypes associated with this. One was here. This individual, a fairly young fellow, actually had a mutation here, and it turns out, which would suggest he might have a heart defect, and it turns out his father died of a heart attack right at the time we sequenced his genome, and he did a stress echo follow-up, and sure enough, he does have a heart defect. He's on medication. So this is all uncovered by the genome sequence. This individual, there were 2 individuals that had 2 X genes. One of them actually did a whole body MRI follow-up, and sure enough, they had early thyroid cancer. Basically, that was removed, and they were to keep most of the thyroid and go through thyroid replacement therapy now. So by catching this, we think that was very valuable. 9 individuals coming into the study were thought to be type 2 diabetic. One of them actually turns out to be have a mutation here. There really what's called a MODY mutation, which is a different kind of diabetes. And the way we treat MODY is usually a little different these days. I guess most people get Trulicity or Ozempic, but certainly back then they were on the suboptimal medication and switched after learning this and got better managed. So the point is that we could actually learn something pretty important, at least from 3 cases, and probably all these people, it's pretty important. They'll get screened more as a consequence of this information. So these are the single gene mutations that put people at high risk. This is typically how people interpret genomes these days. But you probably know that most diseases are not single gene mutations, they're thought to be complex disease. And in general, for complex disease, that's thought to be due to common variants of very small effect. And the idea is that if you inherit a lot of the bad ones, you'll wind up at high risk. If you inherit good ones, you'll wind up at low risk. That's the idea. And so the idea here, again, single gene mutations wind up clobbering you like with a sledgehammer. Whereas these, it's more like, if you get the bad alleles, it's more like death by 1,000 times. So that's the idea. And so the way people actually study these now is with polygenic risk scores. And what they do is they sum over thousands, if not millions, of these single nucleotide variants to make predictions about these complex diseases. And if you look at the odds ratios, it's called, and see how it works, it turns out it works well for the top 2 or 3%. It really doesn't work well for the rest. And we think there are several reasons for this. Actually, does anyone know why? After my talk yesterday, said. Why do you think this doesn't work well for these, the way we do polygenic risk scores? Any guesses? Quiet crowd, huh? What's that? Environmental interaction is probably one, yeah. The other is that the way they actually sum these up is they do linear summation. So they sum these risk alleles. And if you've ever done genetics, you know genetics isn't linear. So 2 mutations in the same pathway, 1 plus 1 is 1. 2 mutations in different pathways, 1 plus 1 can be 15. It can be more than that. And so we think that's a major cause. And the other is they're missing all the rare alleles, which probably contribute to these as well. So we've come up with 2 approaches using AI to better analyze genomes, and I'm going to go through those now. One is for looking at rare variants, and the other is looking for common variants in GWAS types of signals. And I'd say we don't have a perfect genome analyzer, although we know what to do it better. So the first case I'm going to tell you about is trying to look at rare variants for understanding complex disease. We use this for studying this disease called abdominal aortic aneurysm. It's known to be highly heritable, 70%, but only 3 genes have been identified, and they were questionable when we started this study. Basically, we think it's a useful one to study. Phil Sal had a cohort. We think it's important to study because, at least in the U.S., it's a 10th leading cause of death, and a lot of people over 65 will die of this. And the way they figure out you're at risk for this is the aorta burst, and 90% of people die. So it's a terrible way to know you're at risk. Like we want to know ahead of time if we can. So we thought it would be a good one for genetics to try and take advantage of. So what we did was we basically did something that the GWAS folks would be horrified by. We only sequenced 268 cases, 133 controls. We sequenced the whole genome, but we zoomed in on the exomes, looking for pathogenic variants, either likely ones like stop codons or known ones. And then what we did was we basically, from the sequences of these cases and controls, we did machine learning with the tenfold cross-validation. Basically asked for enrichment of genes that were enriched in cases relative to controls. We also, because machine learning can add some features in the electronic health record. And the net result was we came up with about 60 genes with a very high p-value, 10 to the minus 13, that were enriched in cases versus controls. And then when we went in and looked at these cases, it turns out they were sitting in the pathways you would expect: blood circulation, blood pressure, cardiomyopathy for this network. This one's involved in aneurysm. And so we went on to actually look at the mouse homologs of these 60 genes. And they also, their defects, basically, there were knockouts for most of them. And in fact, they gave aneurysm kinds of defects as well. And the genes are both misexpressed in the mouse models and in the AA patients. They all share that. But the bottom line is we think we got 60 genes that are highly relevant. And then we went on to build predictive models for this, where basically the genome by itself has some predictive value. That's the red 1.7. Each R can have some value when you get, especially later in life, when you can measure each scale and such. But together, they're even more powerful. And this one actually does reduce the false positive rate quite a bit. So the net result is we can actually make a predictive test now for this complex disease. It's not perfect, but we have better ways to do it we can do it now. And so the bottom line is I think this has turned out to be quite powerful. We've used this approach for quite a few different diseases, and it started working very, very well. It works well actually in everything, so we've tried. The other approach that we've been working on is this one here. It's trying to use GWAS information and machine learning, and I'm going to go through this in a minute. So this dives a little bit into genetics. And I'm going to tell you about 2 cases. One is ALS and the other is coding severity. We'll get more details in these published papers. So basically, what we did in this case, for ALS, you may know it's known to be highly heritable. It's about 60% heritability. And it's thought to mostly affect motor neurons. So what we did first was we mapped out the open chromatin regions in motor neurons with the TAC-seq and other methods, and came up with something like 100,000 open chromatin regions. And then we said, let's take the GWAS study, where there are over 12,000 cases and almost 24,000 controls. And this GWAS study had found 7 genes by doing standard genome-wide association. But with machine learning, what we can do is for each GWAS signal across the genome, we could ask for the regions that are rich in cases relative to controls for these 100,000, 150,000 regions. Okay, so we're basically, it's a machine learning approach, and once again, it's grabbing combinations of regions, just like the other case. It's very computationally intensive, but it does work because it's non-linear. I forgot to say that's the power of machine learning. It's grabbing essentially enrichments. They don't have to be in a linear fashion. And the net result is it works pretty well. We wound up with basically 690 genes over 7 that were known previously. And we went from 6% of the known heritability to 36%, which is pretty high for those who know about complex disease. Typically, you rarely do you get 20%. And we did find a lot of the well-known ALS genes. We also, there's a list of genes that are misexpressed and such. We're highly rich for those as well. And then we did go on to show that the genes, we call them REPAT genes, they're actually down-regulated in ALS brain tissue, and I guess derived motor neurons. So the sort of thing we would expect. We could go on and characterize the 680 genes. They tend to be enriched in the distal regions of the axon neurons using Go analysis. And then lastly, we went in and took our top gene. The reasons to think there were some strong alleles for this, so we recapitulated them in IPS cell and making essentially these alleles, differentiated the IPS cells into motor neurons. We got out ALS types of defects with reduced neuronal survival, abnormal axon function shown here. And we even have mislocalization of this protein, TDP43, which a lot of people think is causative for ALS. And so our data, what's interesting about this, suggests our upstream factors of TDP43, because our genetics are upstream, if you will, of TDP43, because we knock it out and mislocalize it. So the bottom line is we essentially validated this one, I think, quite nicely. And we're not at a point yet where we have predictive models, but we do have prognostic models. We can tell ALS severity from the genome sequence. I think we now have a good idea how to make it predictive by adding some additional information. So we actually extended this further to start looking at COVID severity. So when people hit 65, their immune system declines, and if they get COVID, there's a good chance to be hospitalized or killed by COVID. But a lot of young people get COVID and get severely, and that's thought to have a genetic component. In general, when young folks get things, people think genetics may be an underlying cause. So we wanted to get at this, and there is some evidence, plenty of evidence, that there is genetics behind getting severe COVID. And so we didn't know what cell types to look for that might be responsible for severe COVID. So what we did here was we actually took all 19 cell types in the lung, and open chromatin regions were mapped out using the single cell multiome analysis for those who follow us. So it's like single cell ataxia plus RNA-seq at the same time, and it maps out all the open chromatin regions for each of the 19 cell types. Each of these is a cell type. And so for every single cell type, you can do what I just said. You can look for enrichment in GWAS signal. Now in this case, there's a very big GWAS study, 1.4 million controls, 5,000 cases, severe cases. And so we look at each of the open chromatin regions for each of the cell types and see if we see an enrichment using this machine learning approach. Once again, combinations of regions that would be enriched, not a simple GWAS study, rather a combination. And so it works. In the end, we came up with 6,600 regions mapping onto almost 1,400 genes. And we look at the genes the cell types that have a lot of this information. Basically, here they are, the 19 cell types. Many of the genes are expressed in multiple cell types. Some are specific to one. And then we can ask where is the heritability sitting in these different cell types. Again, this is getting a little genetic, maybe for a lot of you, but just follow the final upshot of this, which is The bottom line is most of the heritability sits in these natural killer and T cells. We could even go on and do single cell analysis and match to a specific subclass of natural killers as well. So the net result is that actually we could explain a lot of the heritability. It turns out we can explain 77% of the most severe COVID from the heritability of severe COVID from this set of genes. And again, assigning the cell types, these are the two major cell types we think are responsible for the genetic basis of severe COVID. Okay, so we're not yet, same thing, at the point where we have a predictive model yet, meaning from a sequence, can we tell if you're going to get severe COVID or not? But I think we can get there with that as well. So we're working on that. So the bottom line is out of all this is that these new AI approaches let us get at the genetic basis way, way better than we could ever do before. Typically they find 10 to 100 times more genes than they've shown here, either for the rare cases or these common cases. So it's one example of how AI is impacting research. So the other thing we could do is we're tracking people over time. We can actually see how they're aging. And it turns out that everybody's aging differently. So this person, for example, when you look at the biochemical pathway, the top pathway that's changing is this cardiac hypertrophic signaling pathway. This person's a cardio agent. This person over here, a pretty typical age with the coagulation not shown, the metabolic and other pathways change. That's pretty typical over time. We think we need as little as 4 measurements within 2 years that we can tell if people are aging. And so we've classified people into what we call age types, or aging patterns. And we're only statistically empowered to find 4 aging types, but we know there are many more, like the cardio agent. We only have one of these species. And so each column is a different person, each row is a different age type. And so there are 4 people over here, they're aging in all 4 categories. This person's aging in 3 of the 4. This person's a kidney ager, not much else, kidney and metabolic, and so on. So we can see how they're aging. And there are clinical markers associated with each of these aging types. And at least in some cases, people have actually modified their behavior and reversed their aging type. Now, I'm not saying they got younger, but I do think they've actually improved their markers. Okay, so we think this is actionable information. One question I get a lot is, how are you going to scale this up? And the answer is, well, we're in Silicon Valley, so you form a company. And so basically this company we formed, QBio, does a medical version of what I just showed you, but they also do whole body MRI. Okay, so they'll do basically whole body MRI, which is a scan of your entire body. And if you ask any physician today, should you do whole body MRI? At least in the United States, 100% of the time they'll say, absolutely not. Okay. But you know why? What's that?
Costly. Isn't it costly?
Too many. Oh, costly. Yeah, that's one reason. It's expensive. There's another reason too. If I do a whole body MRI on any person here, what will happen? I'll find something. Right? You'll see nodules. We all have them. Men have them in the prostate, women in the ovaries, 100% of the time. We look carefully at it. And if you're asked Mike Snyder, should you do a whole body MRI? I'll tell you 100% of the time, absolutely you should. And the issue is not whether you have nodules, the issue is do you have any growing nodules. And the way you know that is by the longitudinal profile. And so basically for the first 100 plus people, what they found is that they discovered early ovarian cancer, early prostate cancer, even early pancreatic cancer, which is almost never found early. And cardiovascular disease and so on. And sometimes there are multiple lines of evidence, sometimes it was longitudinal profiling, sometimes both, that would uncover these conditions. And so we think, again, it's very important to get your baseline, even with that whole body MRI, because it turns out if you were to get cancer, for example, and this happened to a friend of mine who got cancer. They actually got cancer, they operated it out, and they followed up with whole body MRI, and they found 3 nodules, and they had no idea if they were there in the beginning or if they had been metastasized. So if you don't know your baseline, you don't know how to interpret a lot of this information. So we're big believers it's important to get your baseline information. All right, the other thing we've been working very, very hard on is this idea of Amazonizing healthcare. Okay, the idea, why do you go to a physician when you're healthy to get measured. Now, I think for some things it's probably a good idea, like you're not going to do a whole body MRI at home, those are expensive machines. But you could do a lot at home, and some of the monitoring that we're trying to do is with wearables and this home microsampling. Right? You're taking little drops of blood, mailing it to the lab, and then getting measured. I know what this sounds like, but ours actually works. So the bottom line out of all this is that for wearables, you may know there are thousands of devices out there now, smartwatches, rings, all kinds of different things. How many of you are wearing a smartwatch or ring? Only a few? Oh. Very disappointing. There were more hands up yesterday. So I, of course, wear 8 of these things. Well, some are broken too. You can see my watches, my rings, and that. Even my hearing aid, I do use it for hearing. I need it for hearing. But it measures my interactions, measures physiology. It's a sensor as well. And so these things are very, very powerful, you may know, because they follow you 24/7. And they measure some pretty important things. They measure your resting heart rate variability, and they're mostly pretty accurate for that. They measure other things like skin temperature, blood oxygen, something called galvanic stress response. It's conductance on your skin. Some of them are accurate, some of them not. But even if they're not accurate, the deltas are usually fine. I mean, you can see shifts, again, because they follow you continuously. So we put these on our cohort, started about, I think, about 10 years ago when they were just fitness trackers. And right away we discovered you can tell when somebody's getting ill from an infectious disease from a simple smartwatch and pulse ox. This is actually me when I figured out my Lyme disease pre-symptomatic. Meaning what happened was I was flying to Norway, and on the last flight from Frankfurt to Oslo, what I discovered, you can tell I measure myself a lot, was that my blood oxygen dropped down normally low. It will normally drop on an airplane, so you don't feel the pressure on it. But it dropped way lower than normal, and I measured myself again, and it did come back to normal when we landed. I also saw my heart rate was elevated, it was low resolution. I later saw my skin temperature was up as well. I didn't realize it at the time. And then I did get a fever. This is all pre-symptomatically. And then I did get a fever off and on. So I finally went to a physician in Norway. I wondered if it might be Lyme because I had been in Massachusetts where most ticks were Lyme infested 2 weeks prior to this. So I had a good idea it could be Lyme. And I warned him it might be Lyme. He drew blood, saw my monocytes up, said, yep, you've got bacteria infection. You should take penicillin. I said, no, I should take doxycycline. He got a little tense there because physicians don't like their patients telling them what to do. So he did give in and gave me doxycycline, which cleared up right away. You take it 2 weeks when I got back. I get tested, and sure enough, I was Lyme positive by antibodies, even had some antigens left. And I had given just serendipitously, well, maybe not so serendipitously, but I gave him blood just before I left. I was negative, so I seroconverted during this time. But the point out of all this is I could first I was getting ill before symptoms with the simple smartwatch and pulse ox. So that prompted me to look at basically 2 years of data. And during that time, I saw I was ill 4 times. Once was a Lyme time, twice were viral infections, and the 4th time I had no symptoms, but I had high C-reactive protein, which is a sign of illness. Every single time I was ill, I could see a jump up in my resting heart rate prior to symptom onset. And so basically we wrote an algorithm to detect it. It worked on me, it worked on 3 other people when we got sick twice. Every single time we could see when people were getting ill from a smartwatch in advance of symptoms. So we published this in 2017, and then along came COVID, and did us in 2020, March 2020. And basically we wanted to see whether we could detect COVID with the smartwatch. And if you think about how we do it now, we use, excuse me, temperature. Let's just get a little water here. All right. So anyway, we use temperature, which is a 300-year-old concept. And actually doesn't work very well for COVID, right? Half the people don't get a fever. I didn't when I got COVID. And the other thing you can do is antigen or PCR tests, but they're kind of slow and you don't do them all the time. So we wanted to see if we use a smartwatch for actually detecting COVID, first retrospectively. And so what we did is right when COVID hit, we partnered with Fitbit. We enrolled 53 random people. The 32 people had COVID while wearing their Fitbit and had a diagnosis day and a symptom day. And this is our very first case. This individual was diagnosed here with resting heart rate. They first got symptoms here on day 47, and the resting heart rate jumped up. You can see 9.5 days prior to symptom onset. Very clear signal. So it turns out we could detect COVID in 26 of 32, 80%. And the median was 4 days prior to symptom onset. Okay. So I should say that 80% is for those who were detected before at the time of symptom onset. And then we went on to write a real-time detection system where we take people's circadian patterns. We actually look for a jump up the resting heart rate and some other signals. If the signal's too high for too long, it'll set up a red alert. So we basically built a cloud-based system that can track millions of people. And the idea is just that, track people's health, ideally at a personal level, so you can tell when you're getting ill, if you will. And then also globally, potentially you can use it for tracking pandemics. And so it turns out it works, same number 80% of the time. We have a hard time getting stable baseline uncertainty, but I know with more data types will be better. But right now we're at 80%. And again, here's one of our first cases. There's an individual whose symptoms appeared here. They were diagnosed the next day, and they were getting red alerts for 3 days prior to symptom onset. Okay. Right now, you still get to click on the app to see whether it's a red alert day or not. Signal will send you the signal automatically. But it works on Apple, it works on Fitbit. It should work on all devices because it's an anomaly detection system. And it turns out that it even works in asymptomatic cases. Here's a Fitbit case, works again for Apple on these. Here's an individual who's diagnosed here, but they're getting red alerts for 2 weeks prior to symptom onset. And it turns out it's even more sensitive than an antigen test. Sorry. Let me show you that. Here, which is, this is my case, but we have other cases like this as well. I had a family stress event. I had to travel, and that's like red alerts there. It's mostly green days. We got 1 in 6 weeks is a red day. And then I woke up one morning getting ready to go to New York City. That's a little bit congested. So the question is it allergies? Am I coming down with something? Not sure. So I do a COVID test, and I'm negative. That's the control is negative. So I look at my smartwatch, and I'm positive. What I do, I got on the plane and went to New York City anyway, because I listened to this and not that. And sure enough, in order to go to the meeting, you have to get tested. I was bright positive next day. So I spent a whole week in a tiny little room in a hotel room in New York City, all because I listened to that and not that. So I don't make the same mistake twice. Okay. So it is a very, very sensitive test. We think you need as little as 2 beats per minute elevation, which is easy to pick up, and it'll set off a red alert. Now, I do want to make it clear it's not specific for COVID. Okay, so anything that triggers an increase in resting heart rate, that's the main signal. Other things can contribute too in the algorithms we've written. And the number one trigger, if we look at COVID negative cases, the number one trigger of red alerts is workplace stress. Okay, so there's the mental health aspect of this as well. Other things like intense exercise, medication, most of these are contextual. If you run a marathon, your heart rate will be up for several days. That'll trigger red alerts. Okay, so most of this should be contextual. I got one's triggered. And other infections trigger too, even a mycoplasma as well. So we're actually using this. We're working on algorithms to try and see if we can detect mental health versus respiratory viral illnesses. I don't know if we'll ever be able to tell the difference between COVID and influenza. We'll have to see, but we are trying. We should at least be able to tell the difference between mental health versus respiratory viruses if we pull the data. So we actually, if you have a smartwatch, you're welcome to sign up for the study. Okay. We think this is the tip of the iceberg. If you think about how smartwatches work, they actually measure a lot of stuff. They're shining light right on your blood, and they measure absorbance for SpO2. And so even with this 2016 watch, we could actually pick up clinical measurements. They're not clinical grade, but they're good enough to see shifts from baseline, which follow you continuously. So this is red blood cell count, hemoglobin, hematocrit. So again, decent signals, even hemoglobin A1C and fasting glucose. Again, this is all with the 2016 watch. We think you can measure these. This is useful for anemia and some other things as well. The other area we've been pushing hard on are these continuous glucose monitors. Have any of you worn one of these before? One, all right. So these things are really, really powerful. They measure your glucose every 5 minutes. I guarantee they'll be very eye-opening. So in the United States, 9% of people are diabetic, 33% are pre-diabetic, and 90% of pre-diabetic don't know it. Yet 70% of those will go on and become diabetic within 5 years. And this is rising worldwide. So we're in a diabetes endemic, in my opinion, that's worse than the COVID pandemic, and yet we're really not doing much about it. So these monitors are powerful, as I say. They measure your glucose every 5 minutes. And our claim to fame was we took these and put them on normal people and pre-diabetics. And what we discovered is that some normal people actually have pretty good glucose control. Some are moderate spikers and some are severe spikers, just as bad as diabetics. You can tell the measure. And so we can classify people into glucotypes, low, moderate, or high, based on the glucose levels, right? So the bottom line is, again, we can see all this if you will, in semi-real time using these devices. So we went on to show that different people spike to different foods, as did Aaron Siegel. So some people spike to potatoes, others to pasta, some to white bread, some to brown bread. It's all over the map. It's very, very personal. So here's some work from Aaron. He basically, here's a participant who spikes a banana but not a cookie. Probably the only person on the planet doesn't spike to a cookie. This person's more normal, spikes to cookie but not a banana. Okay. And again, people spike to different food. Here's some of our work. We found some people spike to bread and peanut butter. Those are protein bars, which have carbs. 80% of people spike to cornflakes and milk. You probably don't eat that much here, but in the United States, you know, that stuff's evil. Sugars are bad as you do. Probably worse than smoking, right? Which doesn't kill you. All right, so the other thing, so we've done a lot with this, and so again, I forgot to say I'm in conflict and everything to tell you this. We spin companies off of this stuff. And this company found that just by putting these monitors on people and having people food log with an app, basically they improve what's called their time in range, they improve their glucose control. Because these devices are very, very eye-opening. You'll see what spikes you, you'll be very surprised. And so what happens is both healthy people and normal people will improve. If this person spiked a lot out of range over the last half, just was the first 2 days wearing a monitor, these are the last 2 days. And here's where AI is turning out to be very powerful. This company is basically taking 4 days of training data on people, they have a 32 million food database that can predict people's spiking patterns from their personal habits. And it's very, very powerful. The other area we've been pushing very hard on is this idea of home microsampling for measuring your biochemistry, if you will, from sampling at home. So the idea is that we have these devices, we spent 7 years on this. We collect little samples from your fingertip, or there's another device where you're collecting from your shoulder. You then FedEx it to the lab, and we'll process them and profile them for metabolites, proteins, lipids, as well as some targeted assays on very high-value targets. And so in the end, we measure over 2,200 analytes. And the bottom line is that most of them are stable, so we know which ones are, which ones aren't. There's some lipids that are off. And then we ran some really fun experiments. We had 32 people drink this shake, which is common in the United States. You see it in drugstores and grocery stores. And we profiled people before, 30 minutes, 61, 20, 40 minutes after drinking the shake. And we looked at their profiles. And this is not a surprise. Hundreds of molecules shift. Problems with the molecules at times. And then we watched, and we could follow some pretty key molecules like insulin C-peptide. Here's apolitin, one of these very high-interest molecule these days, even inflammatory markers. And what we discovered is that every person reacts to the shake differently. So these are just 3 carbohydrates. Each box is a different person. For this person, S13, their carbohydrates plummet after they drink the shake. This person's goes way up, 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 up. This person's flat, this person's goes down, goes down. And so we can classify people into different groups, into 5 different categories based on their profiles. And if you look at gray is the average of the group. For this group of people, brown are inflammatory markers. Let's focus on those, quite interesting. For this group of people, their inflammatory markers goes down when they drink the shake. For this group of people, it goes up, same with that. Same shake, pro-inflammatory on some, anti-inflammatory on others. At least in the United States, 10% of people have irritable bowel syndrome. They have no idea what's causing it. We could actually measure it in this case to see what's going on. So we think, again, these are pretty powerful technologies. And then here's what I see as the ultimate experiment. There's one individual, you can probably guess who it is, basically gave a sample every hour for 7 straight days, 98 samples total during waking hours, not night. And then also, while wearing a glucose monitor, smartwatch, food logging, measuring activity. And over 1,000 molecules changed. And then what we do are these time-like correlations with the idea that cause of events lie upstream of downstream events. And so it doesn't prove it, but can suggest that. And so we look for these associations. We're trying to see associations between people's biochemistry and physiology with, say, their activity and foods they eat and things like that. And so the net result is that we find literally thousands of associations with, these are, so here's heart rate, glucose, and steps. This magenta color are the cytokines, the inflammatory markers. These are lipids, and green are metabolic. So we'll see all kinds of associations, and many of them are extremely interesting. So we'll start with the simple one. Your insulin C-peptide goes up after glucose. That's not a surprise, but we can measure it. It's 10 minutes in this person. Here's a very interesting inflammatory marker that also goes up in response to glucose, transcription factor, which we think might be responsible for the inflammation that occurs in diabetics. Here's a really interesting one, alpha-synuclein, which is involved in dementia and Parkinson's. It also shows an interesting pattern that we think correlates with stress. So imagine you're high risk for dementia and Parkinson's. You might want to know this, and then try to mitigate the behaviors that would cause these spikes that perhaps you can push off dementia or Parkinson's. That's the idea, something we're testing now. So we think these kinds of, personal markers can be very, very valuable for modulating behavior. So what we've done now is we have a dashboard that can pull in your wearable, clinical, microsampling data, and display it in whatever resolution you want, daily, weekly, monthly, what have you. And then ultimately what we'd like to do is bring this into a healthcare system. And this is what I see as Mike Snyder's world, not everyone's world, but I like the idea of people getting the genome sequence before they're born, which is now possible. And then together with detailed biochemical wearable measures that will track people and build personalized AI tracking systems, which you can do around people that can better manage their lifestyle and their health. And so ultimately we should be able to better predict risk, diagnose disease early, monitor and treat disease with big data. And I will tell you that, again, the way we try to scale this, I think academics are really good at basically discovery and proof of principle. We're not very good at scaling. So what we do is try and spin off companies for this that can actually scale this. And this is our CGM company. This is the microsampling company. It'll take pricks of blood and give you back a report on about 600 metabolites. And the net result is that for this, as an example for this one company, basically the metabolites fall into several wellness categories, if you will. Things like you can get information about oxidative stress, longevity, inflammation, so on. We can even make predictions or calculations of people's biological age and see if they're younger or older than the chronological age. And here's a fairly typical case. An individual happens to be older than their chronological age, the biology is older. They're fine on inflammation. That you can tease out the age detection. They're fine on inflammation, not too bad on metabolite age, but their heart age is older than it should. And so using AI, you can pull in information and recommendations on lifestyle changes these individuals can take to improve this. 95% of people actually improve their markers after getting the test results. So we think actually this could be very, very powerful for the future. All right, so that's the story I wanted to tell you. I have an amazing team that's done this over the years. This is my wearable scheme. What is that? It's big. It's not that big now. Half a dozen people. Here's the microsampling group. Shout out Shannon, Ryan, leading this. A number of people have led this over the years. So I think I saved a little bit of time for questions. If you have any, we'd be happy to answer. Thanks again, Pat.
So for the first part of your talk, you emphasized the importance of the genetic basis of disease. My simple question is, have you ever tested the difference between different ethnic groups in your model?
So the answer is not exactly, no. For the single gene mutations, they're mostly based on European, although many of them, like BRCA, go across. And for the GWAS data, it's unfortunately mostly based on Europeans. So it is shifted, unfortunately, but there's a big emphasis in the United States in general, and then we have certain projects that are actually focused on underserved groups that are actually drilling into some of these signals. We're trying to get signals basically from other ethnic populations. You might be pleased to know that for our IPOP cohort, 109 people, it's overrich for both East Asians and South Asians, relatively, but it's still more Europeans than any other group. And it's also still a small group. Now we've got 171, but we have many more. We'd like to get this very large, but now what's happened since we started this, many large-scale projects have popped up, like the All-of-Us program, the UK Biobank, are now running this. They have much bigger budgets than we do, but this is just one lab trying to scrap it together. So I hope that we'll get this much broader. And I learned that you have a nice biobank here as well. That will be probably fantastic for Taiwanese, I would assume.
I think the audience needs to know you haven't changed much since the Yale days. We're going to play softball together. For the question, so ethnic is one. So in terms of demographic, so have you seen any dependence in terms of the demographic within the states? Because it's East Coast, West Coast, and middle of nowhere. So are there any in terms of diet and environmental cues that can probably extrapolate to other?
Yeah, great question. So the answer is this is mostly Bay Area because we're very careful about our sample prep.
Yes.
So if you are from Southern California, believe it or not, you fly in to get measured. But in general, it's people from the Bay Area, and it's a somewhat overeducated group than average, although it's reasonably diverse. As I say, we do have a lot of Hispanics, we have some African Americans, as I say, a little bit over, not a little bit, a lot overrepresented for East Asians and South Asians. But it's still a small sample size, so we're not really powered to tease out ethnic signals, if you will. We do have a project where we sample people from around the world, and there we are teasing out ethnic signals. It's kind of interesting. But we do track diet and all those things. Then we do fun things with diet. I left this out, but we can look up a very cool paper we did. You've been told, I don't know what it's like in Taiwan, but we're told since we're little in the United States, that there are 4 seasons and they're 3 months each. So we just took the data we had and said, well, why don't we see how many seasons there really are? What does a biological fluctuation look like on a calendar year? Are there 2? Are there 15 seasons? Who knows? And why would they be 3 months each anyway? That's just an artificial construct. So we actually looked at the data, and you look up the paper. It turns out there are 2 seasons in California. There are 2 major patterns, and there's some sub-patterns, but 2 major patterns from the cohort we have. They said, well, it's California, that makes sense. But they don't quite match the way you would expect. One is a big peak in winter, and that correlates with viral infections and some things we don't fully understand, so that makes sense. But the other peak, in hindsight, makes sense. It's like late April, early May. And that one correlates with allergies and asthma, so that also makes sense. So those are the 2 major seasons we would argue are in California. They're not 4, they're 2, and the peak of 1. That's kind of interesting because you can see cardiovascular patterns for the groups who are doing this sort of thing. A lot of very interesting data in those seasonal patterns, and we totally ignore that in today's world. Now we should build that into health models.
Really
long answer for your.
Yeah.
You hit something I'm kind of excited.
Really quick question. In terms of wearables, you have 8 of them, right, so far. Is there a in the future world in terms of like one personalized wearables that measure in terms of not necessarily everything? Obviously, you have like different. You have like health watch and stuff. So can you imagine like or comment on in terms of one personalized wearables that linked to a database or linked to a personalized throughout life, something like that.
Yeah, if you're going to get the one, you probably want an implantable, because then you can measure biochemistry like glucose and things. Right now, you measure through interstitial glucose. You want to get heart rate and all those things too. So if I were to only have one, I would probably want it to be an implantable. But I think we can live with two if we had to, if we wanted to do a regular watch. And the reason I wear a lot is because there are different resolutions and measure different things. It's for science, not for, because I like wearing these things. It's just, yeah, it's for measurement purposes. So I think we'll pull this, yeah, this is science. So I think the world is going to be exactly as you say, you know, where we have dashboards for this. And so right now, the analogy I like to use, you know, your car has tons of sensors on. A race car has at least 400. And they relay that information back. Your own car probably has at least 20, if not more. And it relays it all back to a dashboard that's pretty simple for you to interpret. And you know, you have speedometer and gas, it's an easy one, but you have a check engine light, which often has a lot behind it. And so you get that light on and then you do follow-ups. And that's how I see this all rolling out in the future. That we'll have health check engine lights and it'll probably be your smartphone. And you can decide whether you want it to buzz at you or not. But that can determine, you know, how you would act on that. And that's what these are. They're alerting systems that you would then go back and do follow-ups on, much like your car. And I find it kind of ludicrous that we have all these sensors on our car, yet none on our body. We count on our internal ones, which are, you know, questionable at times. And so I would argue our health is probably more important than our car. So we really should have personal health dashboards.
You showed that the analysis can give you many more genes than previously identified, like the ALS 6, 9, and 8.
Right?
But these have to be functionally validated using animal experiments, which are expensive, slow, and it's basically a bottleneck. Do you see how to get around this? Could it be at some point that we have so much confidence in these data analysis, we can just ignore the animal study without knowing the fundamental mechanisms?
I hope so. It's nice still to know the mechanisms anyway. The way we do a first pass validation, first you try to repeat it, so we have a separate cohort. I didn't get all the details for ALS, and got basically like 490 overlap with the first set. So we feel confident most of this is right, the second one is big. And then you can do things like the misexpression I mentioned, and I did a quick flash of that. So you can find things that will tell you you're on the right track. And that's what we do. We do a lot of network analysis and then see where those networks operate. Like I mentioned for the AAA case, they were in blood circulation and things like that, which is what you would expect for AA. So you can use all the information that's out there. But you're right, ultimately, if you really want to get in more detail, you knock them out. But if you don't get a phenotype, you're not sure necessarily. We like to knock them out like ALS, we do in organoids, because there is a phenotype you can follow. But if you don't see a phenotype, it doesn't mean it's not an ALS gene. It just means you might not be in the right conditions for it. So anyway, and we know ALS actually gets accentuated with exercise. So there are certain things you might have to do to tease out phenotypes you have. So I think, you know, I use a lot of this other sort of circumstantial data to kind of validate first. And that's kind of low cost because the data are already out there, or you can do expression data. That's not hard. But you're right, the knockouts are pretty laborious. But it is what we're doing as a follow-up.
Yes, very comprehensive speech. I have one comment and one question. For those patients who have diabetes, we are seeing many using the continuous glucose monitor here in Taiwan. It's our pocket money, but still we see many patients. But as you said, that we expect to see more proportion to reach, so TIR, target range.
Right.
But we have to emphasize telling the patient that it will not become increase your TIR until you change your behavior and medication based on your findings of those curves. So someone believes that when I'm wearing the CGM, then that glucose becomes better. Is that right?
Yeah. What I think is nice about the glucose monitors, it is very visual, and it really does hit you when you see what's going on. An example I like, and the fact that people improve their TIR, we didn't tell them anything, there were no recommendations, they did that automatic. And but if you do wear these, you can see why. Like there's a New York Times reporter who basically called me up and said, you know, I used to have I thought it was the healthiest lunch. I had salmon on salad every day for lunch. What could be healthy? Then he puts on a CGM, and his glucose spikes through the roof. Guess what it was? The sauce. There was sugar in it. So all you had to do, what you did then, was leave off the salt. You got the healthy lunch you wanted. But some of the stuff was obvious in hindsight. For me, it was a pulled pork sandwich. So I ate a pulled pork sandwich. I watched my glucose. Now I'm type 2 diabetic. I forgot to say that. So I went to 350. And I'm like, look at this thing. I'm like, what's going on? I showed it to a friend of mine. He goes, Mike, everybody knows pulled pork has sugar in it. I didn't know it. So some of the stuff is very obvious in hindsight. And I'll tell you, I don't eat pulled pork anymore unless I know it doesn't have sugar. So you do learn a lot from these. It's so visual. And I guarantee any of you who wear one of these things as a spiker, you'll never eat the same. In a good way.
Yes, also I believe that CGM will be widely applied also to those pre-diabetes.
We do, yeah, and I think that's
an
the key
obesity.
to prevent diabetes. Yeah.
So the question is, I heard your speech of the GWAS, polygenic risco, metabolite application, but how about the whole genome sequencing, those information that are available? It's become cheaper now, right?
Yeah,
They're
I mean,
linked together.
Yeah, we tend not to do, we're using existing GWAS studies. When we do a study, we actually always do sequencing. So we can try and use both approaches. If we sequence enough people, we can see if we get a GWAS signal, but then we can also use the rare variants. And I hope in the future we're going to integrate both of those into our predictive models. That's the goal. We're not there yet. There's some tricks that would make it a little bit not as straightforward as I'd like to hope. But anyway, yeah, I do think it doesn't really make sense to do genotyping arrays anymore. Sequencing may have $100. So we might as well just get the whole sequence.
It was an amazing talk, and I used to be a clinician, so I think the COVID wearable data is fantastic because it is a difficult disease to diagnose from symptoms. And if the heart rate is so, and that's one of the most reliable digital data to convert for machinery and all that, how would you get that information so that it becomes a clinically relevant, usable diagnostic tool? Because we are going to face, infectious, disease. We see that now. So how would you get that into the clinical?
Yeah, so you have to run a proper study. This
have
is a research study right now. So the devices there's a certain approval process you need for that. So some of them have that, like Apple and such. And then to actually turn into a study, you do need to run the proper study with the algorithm
just like a
and
typical,
prospective
right?
study. Yeah, and we will, again, it's always going to have other non-COVID signals associated with it. And I think you just accept that. These are real stressors. Whenever you get a red alert, it's not like, oh, those are background noise.
Right.
They are from the COVID standpoint, but they're really, if your heart rate's going up, in fact, you said it precisely, when you wake up in the morning, your resting heart rate is very, very consistent. And if it's off, you're either mentally or physically stressed, one or the other, guaranteed. So we just need to run a proper study. And of course, they'll want to show utility, that when you do that, you actually, the studies we're running now, to be honest, are all transplant patients. Because if they get an illness and immunosuppressed, they can die. So we have smartwatches on a bunch of people who are transplant patients and trying to see what the frequency of detection is, relay things back. And then even if it's not COVID, we have, there are other agents you can use. I forgot which one. Well, it's with a particular company that has their own Tamiflu equivalent, if you will, that people can take. And certainly if it's for COVID, we think you would have Paxlovid, in your home dresser that you would take that right away if you see a red alert, where it might be more effective. So I think those are the kinds of trials we need to show efficacy, and then hopefully it would get adopted. But yeah, I think a lot of people are sort of winging it because a lot of people do with smartwatches. So it's pretty clear. I mean, I've had people come up and say how they heard my talk and 2 months later or something went off, and sure enough they had strep or whatever. It's kind of interesting.
Sir, I apologize. I have bad cold, so I think the voice is clear to you. Okay. I would like to think this precision health in the perspective of precision health. In the public health context. So when you're using these different wearables and microsensor, what kind of your clients or individual, are they connected to the primary physicians and medical center? How are they being consulted when they have some come up? That's the first question. The second question also related to public health. Would it make more sense do you consider to do prioritize? Do you have some genomic data and then therefore you can guide someone to use certain wearable and vice versa?
Yeah, great question. So everything we're doing right now is a research study. So it's not tying into clinical, although several groups have reached out to us. We are trying to do some clinical monitoring and working with physicians in very specific cases like in ER and other cases, trying to bring in these wearable data to see if we can basically track people. And there's a lot of interest in using this for outpatients, where people, they want to get out of the hospital. There's plenty of studies say people recover better at home than they do in the hospital. So if we can track people outpatients, that's probably a great place to start. And so we're trying to set up some studies in that. And there's a lot of discussion about this sort of thing, what's happening. It was getting implemented as, it's in progress. The other, the second thing he used genomic data to predict what wearables and things. We're not even close to that yet. So, but we are trying to integrate omics data with wearable data to make better models. And the nice thing about machine learning is all these things are features. The omics data are features, wearable signals have lots of features on them, like the shapes of the curves and things like that. You can pull those features off and then basically build machine learning models for conditions you're trying to detect. And so they're very, very powerful. The combinations that are more powerful than individuals alone. So we're trying this for a lot of different things, for diabetes and other situations as well.
Thank you. The second short question, is any of these items reimbursable by the U.S. Health insurance system?
For healthy people, the answer is absolutely not. And that's the number one problem, because our whole system is broken. Going back to this, nobody pays to keep you healthy. Okay, and that's the number one problem. You have to, everything I showed, like QBio, all those companies I put up again, you have to pay out of pocket for that. Even if it would save the health system money, it just doesn't work that way. So the head of the hospital said, Mike, nobody pays me unless they walk in the door, and they're not walking in the door unless they're ill. So it's a real problem because our whole financial incentives aren't aligned properly. I don't know about Taiwan. I think single payer system have a much better chance of implementing this stuff because they will, I think, save money in at least certain circumstances. You can model it out. Like if you're in a family that has cardiovascular disease risk, you should get your genome sequenced because if you get a stroke or a heart attack, you can wind up with long-term disability. It's very expensive. And in the U.S., one day in emergency care is $50,000 or more. So basically, we can make arguments for this to be the case, but unfortunately, the system's not set up that way. So we really need to remodel everything. And so, I think there's only two ways, as I was saying yesterday, to motivate people. One is money and the other is family. So if you could give people a discount if they were smart blocks or got their genome sequence, maybe that would incentivize people to do this more, and therefore hopefully keep them healthy. So the goal obviously is to extend people's health span, live healthy, and then just stop. Hang on too long, not too great state.
Simple question. So you run a lot of monitors on yourself.
Yep.
Did you have to go through IRB?
Well, believe it or not, I did enroll myself, and I was very general on it.
So that's
Just to play it
comprehensive.
safe. Technically, you don't have to, though, for self-experimentation.
The people in your lab cannot involved in the experiment, right?
They're not supposed to be, correct?
Yeah, we okay.
You're all okay, yes. So people in my lab aren't supposed to because they worry it's coercion, that I would be coercing people to actually do it. But of course, people in my lab would love being in studies, and I'm sure some simple way, and I don't know, under pseudonyms or something, I don't know. Anyway, yeah, so in the United States, you can't do that. So if you want to be in one of my studies, you shouldn't join my lab. Counterintuitive, right?
Okay.
All right, thanks again.
Next year, we're going to invite Xiaotao come here, give the talk. Xiaotao is moving to Singapore in May, also very outstanding postdoc. We have some souvenir for you. Don't walk away. It's good for your work.
Wow.
Every day.
Wow, that's huge.
Every right.
All right.
We're going to see the genome sequence.